How are we treating patients with COVID-19? What are the stresses on doctors? What are some of the things that doctors have learned? What are some of the more meaningful experiences that doctors on the front lines have actually had treating this disease? And all of us, all three of us, have been personally involved with patients with COVID-19 in our practice. I can say that with certainty. I thought we would start there. And then we'll move on to some more, the next sort of set of questions that I think people had regarding what this infection really is and why certain people appear to get certain manifestations of the disease more severely than others do. And then I thought we'd end by talking about questions of treatment, vaccines, testing, and things that I think a lot of people had questions about as well. So, and then next week, just to sort of give a little bit of a preview, we have some experts coming on who will probably be best to talk about the last little set of questions that I've gotten so far, which is really about the nature of the pandemic itself from a public health perspective. How it began, when it probably began, how it got here, how it's progressing, and how it ultimately would end. And I think we'll get into more of those kind of discussions next week. So why don't I start by first asking some questions here. First of you, Dr. Marmor. I think, and for those who don't know Meghan Marmor, she's an assistant professor of medicine in the Division of Pulmonary Medicine at Stanford University, and has been particularly important in Stanford's response to the surge planning that's been required for the COVID-19 pandemic and for taking care specifically of COVID-19 patients, both in the hospital and in particularly in the critical care units. So she's been integrally involved in both the planning and carrying out of the care plans and the workflows for caring for patients in this trying time. So first, I'd just like to start with some general questions, Meghan. What's it been like taking care of patients in the ICU? Stanford had a lot of patients in the ICUs. Just give some general numbers of how many have been there, how many are ventilated, for example. Everybody's talking about ventilators. What has been the survivability of patients, and what is it like actually taking care of patients on the front lines?
Absolutely. So hi, everyone. I'm Meghan Marmor, and just so that
Wait, you're muted. You're muted. Hold on.
Let me try talking. Can you hear me?
Gordon or Peter, do we need help unmuting?
It's all good. You can continue talking. We can hear you.
Oh, you can hear me? Okay.
There we go. Okay, you're good.
Okay. So that just so folks know and understand what I do. I am a lung doctor who also takes care of people who go into the ICU who are critically ill, who need to be kept alive on life support using things like a breathing machine. And so far, to sort of jump right to our question, our numbers have been fairly low here. So right now, as it stands, we have about 13 patients at Stanford Hospital at our main campus, and then a total of 7 patients at one of our associate facilities in Pleasanton, California. We have 8 patients in Palo Alto on mechanical ventilation in the ICU, one of whom has passed. We have a total of 6 people at the Pleasanton ICU, 2 of whom have been able to discharge and leave the hospital and are entering a phase of recovery, and no deaths at this point for now. So it's been really interesting, since this all started, and it seems that starting somewhere in mid-March, all of our lives suddenly changed dramatically. And in the same way that our lives had to change, it was as if our entire organization, our entire hospital, all of our clinics, and everything that we do had to be completely revamped, top down, in a way that would allow us to continue taking really good care of patients in the way that we know how, in the way that we want to do, while still respecting and making sure that we ensure the safety of everyone who comes into work. I can tell you that after 4 weeks of what feels like endless meetings and instituting new protocols and developing and changing and adjusting those protocols, I would say that as an organization, and certainly from the ICU perspective, I think we've come up with a really nice system. So for instance, we've been able to isolate and establish a corner of the hospital where these patients are kept and are taken care of. So we have dedicated staff and dedicated rooms that are just for COVID-19 and are separate and distinct from the care of all other patients. The staff who enter those rooms don't go over to other rooms for the segment of time that they're serving, and then they have a break period before they go back to taking care of patients who do not have or are not suspected of having COVID-19. We've implemented a number of safety measures to make sure that we can access personal protective equipment and keep our stores up so that we don't burn through that. So as it stands, I would say that coming to work, I can say from my own perspective, from the perspective of many of my colleagues and a lot of the nurses and respiratory therapists that I work with, a lot of us have commented, we're feeling really safe right now because we have been able to maintain our health. We've been able to access personal protective equipment. And so far, things are going pretty well. We've actually created a number of different guidelines on how do we, if you need to come to the hospital, how do patients who suffer from COVID-19 or are suspected of having COVID-19, how do they come into the hospital in a safe way that protects staff and protects any other person from the community who needs to also come to the hospital so that there's not close contact between those groups? How do we then move a person from the emergency room up to an ICU in a way that protects everyone around them? And so we've actually come up with a really nice system that I think has set at least our minds at ease.
Are you masking everyone regardless of the reason they're coming into the hospital when they first come in through the door in the ED? It took us about a month in the hospital to finally figure that out before we started masking all employees and all patients before they came in. And then the second question is, are families allowed to come in and visit with patients as well right now?
Great questions, great question. So yes, we have implemented masking for all people, all staff, and all patients actually, if they're not within their room, and especially if they have suspicion of COVID infection or COVID positive. You know, I wish I knew, and I wish we could say with any kind of confidence how transmissible is this virus from a person's cough or even just from regular breathing. And I honestly can't say that I know, and I don't, and if somebody were to tell me that they knew the answer, I'm not sure I would believe them either. So all I can say is I know that I feel more comfortable knowing that my face is covered. I believe that it offers me some degree of protection. I'm not confident how much, but I do think that by having everyone cover their noses and mouths, I think all of us feel a little bit more at ease that we are doing something to curb the transmission of that virus. In terms of visitor policy, that has been such a painful thing for a lot of us because we know that this is a virus that is transmitted between people, and the closer people are together, the more likely it is to be able to spread. So as a general policy, we have severely limited the ability of people to come and visit the hospital. Really, we only make exceptions for those who are nearing the end of life, and they are only able to really have up to 2 visitors, and those visitors have to wear full PPE. And then for more special circumstances, if a person weren't able to normally advocate or speak on their own behalf, if they need a representative to come in, and that requires that person to be present physically in the room, and it can't be done via phone call or via video like this, we do make some exceptions. But that's been pretty heartbreaking. I mean, I would say that if my loved one were in the hospital and certainly on a mechanical ventilator, I would not want to leave the hospital, and I would want to be with them all of the time and know that as your healthcare team, and I think I can speak for everyone when I say this, we wish you could be there and we want you to be there. And if there's anything that we can do to facilitate that, we're trying.
So I have numbers for San Francisco General. Erin, you might know them for Parnassus, and I could probably look them up too. But to date, in our ICU alone, not in the hospital per se, I know we've taken care of roughly half of San Francisco's COVID-19 hospitalized patients at San Francisco General. And we've taken care of 38 patients in the ICUs alone, 24 who ended up being intubated and put on a ventilator. And over the last couple of months, 17 of those patients have been discharged, and we've had no deaths right now. I know Parnassus has had 2 deaths. We've had none. So we've had 38 people get sick enough to be in the ICU, most of whom have ended up on ventilators, who've ended up surviving. And I wanted to know, I mean, obviously this is something that we're learning about on the fly. It wasn't, again, we don't have the luxury of a 100-year history of knowing this disease and understanding its characteristics and knowing how to treat it. So a lot of what we've learned about it, or are learning about it, is being learned on the fly. Meghan, can you tell us a little bit about some little tricks of the trade and interesting observations that you've noticed and that other clinicians have noticed both at Stanford and around the country that have led you to improve the care that you're giving to patients as time goes on during this pandemic?
Spotlight your video. Unmute myself. Okay, can you hear me? Okay, good. Okay, so I have been very surprised by who we've seen come in and who has gotten horribly ill and who has cruised through their COVID-19 infection unscathed. I will say there have been, I think of 3 individuals who stand out in my mind who 3 months ago, to their knowledge, were completely healthy, who came in and were discovered to have diabetes that they didn't know about. It was very poorly controlled, and they were morbidly obese. And they have had a very difficult time fighting this virus and have been quite ill. We've then had a handful of patients who have known chronic lung disease, who I would consider to be a person who has chronic sick illness. And they've breezed right through. I had a video visit with one of them and they were having a beer on the couch. So who has gotten very sick and who has not has continued to surprise me. And so I'm keeping a very open mind now. I think I went into this virus thinking, oh, okay, if you have these specific illnesses going into the virus, you're likely to have a really difficult time with it. And now my attitude has really changed where I can't say consistently that I know how each person would deal with this kind of infection. I think the only other thing that has sort of come up that I've been grappling with is, you know, these patients require quite a bit of oxygen, and how we deliver that oxygen. Do we want to move towards the mechanical ventilator earlier, or do we want to wait to do that later? And I don't think for right now as a field, we have a really clear answer on that, and who is the perfect candidate for mechanical ventilation and who is not. So we're still learning a lot about this disease.
Yeah, I'll share a couple of personal anecdotes that we're writing up right now. I have patients on very, very powerful immunosuppressive medicines to treat their autoimmune diseases, very, very powerful biologicals and such. And I haven't had anyone. I've had several that have had COVID-19, right? Including some that have been sort of unprotected, sort of right in front of my face, because they come in with very atypical, very banal kind of symptoms that aren't classic for COVID-19, and several of them have been probably viremic for 2, even more months at a period of time that their symptoms have lasted until they finally been diagnosed. So clearly patients are behaving in ways we don't understand, and the immune system is a part of that. So to Erin, to you, why don't I transfer a little bit and talk about that very important question. We've had several people, I'll even read you, so give you a flavor of some of the questions. This is from one congregant. We are told people over 60 are particularly vulnerable. To what extent and why are older people at risk? And if they're healthy and have no specific risk factors such as diabetes or obesity or respiratory issues, et cetera, why are they getting sick? And so I guess the general question, someone else said, for someone in the lower end of the senior range and otherwise good health, how much greater risk do they face of being seriously affected relative to the population? And so the question for you is, how does the virus typically establish an infection in the respiratory tract of people? And why, or what are some general thoughts about why it is that certain people with underlying medical conditions, and particularly older people, are really vulnerable to getting the worst manifestations of this disease?
Okay, wait, can you hear me? Jonathan, can you hear me?
Yes, we can hear you.
Yes, we can hear you. You're good.
All right. My child walked in right at the inopportune time. So before, yes. Okay, come sit on my lap. So before I answer that question, I want to highlight two things. One is that people shouldn't stay away from the emergency room because they're afraid of COVID. The ER at UCSF is particularly light, and we have established protocols for keeping people who have respiratory symptoms away from other people who don't have respiratory symptoms. So if you're having symptoms of a heart attack or stroke along those lines, please call 911 and go to the emergency room. We don't want people staying at home because they're afraid that the ER is too busy. The second thing I wanted to just briefly touch on is how this disease has disproportionately affected an underserved population, and really calls on us as physicians to advocate for people who don't have a voice, people who are homeless, people who are housed in very crowded living conditions, people who are on the front lines and have to go to work, even though they prefer to stay at home. So that's what I wanted to say before I said anything else. And then, because I don't have an answer to your question, the answer to why, this is a new disease that we don't totally understand. The virus is one of the largest viruses that we have. So its genome is very, very long in comparison to other viruses. And much of the RNA of that virus encodes proteins that we don't know what they do. So we think that much of these viral proteins hijack the host immune response to cause the kind of severe illness that we are seeing. But why it affects some people and not others is really unknown, and I would just be speculating. Now, I can speculate as a scientist as to why that's the case, and I think that's what people probably want to hear, because this is really kind of an exciting thing for scientists. So I'll just tell you my speculation. Why elderly folks are at greater risk? We know that we have basic science studies that show there is a particular cytokine that is absolutely required for clearing viral infections from the upper airway, and that is interferon. And we know that elderly people produce less interferon than young, healthy people in response to viral infections in general. So this is one of the reasons why you can get reactivation of viruses like varicella on the skin in elderly people. So we know that elderly people have this as a risk factor. Now, why are we seeing young people who have diabetes or high blood pressure developing very severe cases of ARDS? We have a bunch of reasons we speculate why this is happening, and much of them, much of the hypotheses revolve around the immune system being overly stimulated, or stimulated in a way that is very, very exuberant, and we call that a cytokine storm. And there are a number of drugs that are being tested in randomized control trials, both here in the United States as well as in Europe, to test whether these drugs that can block this over exuberant immune response can improve the outcomes in these patients.
That's great, Erin. So
the last thing I'll say is this issue of genetics. And so I do genetics in my laboratory at UCSF. I study asthma genetics, and the thing that is, asthma is a very heritable trait, meaning that if your mom and dad have asthma, you are a much greater risk of developing asthma. But what a lot of us forget, because we have lived in an era of antibiotic use, that the risk of death from infection is also a highly, highly heritable trait in a pre-antibiotic era. So there are very common genetic mutations that we all carry that confer an increased risk of dying from infection.
I'm so glad you mentioned that. That's, I was hoping you would say that. So genetics are probably a very, very important part of this. Let me just review a little bit of what you said for the people out there so they can understand. So when a host cell is infected with a virus, right? Viruses, my wife and I had this conversation a month ago where Jessica was asking, are viruses living things or not? And I said, well, it's debatable, but generally people believe viruses are not living because they can't replicate on their own. For example, the COVID-19 virus, the virus that causes COVID-19 is an RNA virus. It doesn't have DNA in it, which is the foundation of life, right? So viruses are required to infect cells, almost like a parasite in a way, and hijack the host cell machinery to replicate itself, right? It requires a host. And so one of the difficult things for the immune system is to determine what cells are actually infected and what cells are not, because the virus lives primarily intracellularly, inside of the cell, right? And so when a cell gets infected with a virus, it has to raise its hand and notify the immune system that, hey, I'm infected, help, right? And the immune system usually responds and says, I'm sorry, pal, but if you're I can't extract the virus from you and let you live. So often what the immune system will do when a cell announces that it's infected is come in and basically kill off the cell and hopefully the virus that's sitting inside of the cell. And one of the ways a cell raises its hand and says that it's infected is it releases a bunch of what we call cytokines, some of which are interferons, the family of interferons. And interferons are what enables the body to then mount a certain specific kind of immune response that will kill the cells that are specifically virally infected. And there's some data, data from animals and certain kinds of infections, particularly with other coronaviruses like the original SARS virus and the MERS virus, that suggests that if you're young, it's kind of like the three bears and the wolf and the porridge, right? That if your porridge is too hot or too cold, that's not good, right? If you have too much interferon, if you have a very robust interferon response, that maybe you get a very aggressive immune response to that can end up not only killing the cells, but creating collateral damage as well, which is this cytokine storm that Dr. Gordon was alluding to that leads to people becoming sick throughout the organs of their body, including their lungs and elsewhere, and ultimately dying. If you don't have enough interferons, as was speculated by Dr. Gordon, say if you're a little bit older, it allows the virus to sort of take hold and replicate more and more and more without the immune system coming in and punching it out, because it's not getting as aggressive a response. And that may not be such a good thing either, because it allows the virus to take hold and replicate and not be brought under control. And so what you really want is the just right response. You want the right amount of interferon response and the right amount of immune response that takes care of the viral infection where it is without damaging, having a lot of collateral damage. And I think that's a lot of where I think this research is sort of going to sort of answer those questions. Can you talk a little bit about research going on right now, Erin, both at UCSF? I know Carolyn Calfee and others have some very interesting projects, the COMET study and stuff. There's a lot of stuff going on right now that's giving hope, right, to understand a lot of these questions about why the virus behaves the way it does, why some people are getting sicker than others, and is there any way people can help if they want to help?
I think the easiest way that people can help is through an app on your phone called Eureka. And you can go to the App Store and download it. It's part of a citizen scientist initiative that a group of investigators who were really doing research in cardiology devised. And once the COVID-19 pandemic came about, they changed the question algorithms to be focused around COVID-19. And so every day, if you download this app and you register for this clinical trial, it will ask you about your medical conditions and the drugs you take. And every day it will send you a notification, and you can go in and answer about 3 questions: whether you have a fever, whether you have a cough or shortness of breath, how many people that you have been in contact that aren't inside your family unit, and data from all across the United States, as well as in Europe, in China, all across the whole world is being collected. So citizen scientist, and the app is called Eureka, and I do it every day. So that's probably the easiest way to help do research. The easiest way to help us do our jobs, what I think Megan would agree with me, the easiest way to help us do our jobs is by wearing a mask when you go out and continuing to socially distance yourself, because that will maintain capacity at our hospitals to be able to take care of you if you get sick or if your loved ones get sick. For the research that's going on at UCSF and other places, there is so much happening that it is hard to keep up with. So we have a system of presenting research in the United States that is usually through peer-reviewed journal articles. And then there is this other way called the bioarchive. So peer-reviewed journal articles are when someone does years of studies and they write up a journal, they write up their article and they submit it to this journal, and then the journal takes your article and sends it to 4 or 5 other investigators, and they all tell you what's good about it and what's bad and what they want to see fixed. And then you go back to the drawing board and rewrite your article. And that whole process takes a long time, but it's great because that peer review allows the best research to get out there. But it's slow. We have this other system called bioarchive, which has been around for maybe 5 or 6 years, maybe even longer now, in which you can just post, any scientist can just post their research article, and it isn't peer reviewed, and they post it there in hopes that it can disseminate the research faster, and people can learn more quickly. Well, bioarchive is getting extraordinary numbers of posts of articles. In fact, like hundreds and hundreds a day. I cannot keep up with it. So there is so much research going on. In fact, UCSF has hundreds, and Stanford as well, hundreds of research laboratories, and they have all been shuttered during the shelter in place. And the only research that's happening on those in those laboratories is COVID-19 research. That's the only thing that's allowed to happen. So my lab is doing some very small studies on COVID-19. And other labs are doing those. We have clinical trials going on in the hospital, so does Stanford, where we're testing drugs in patients who are hospitalized but not on a ventilator, and in patients who are hospitalized but are on a ventilator. We have studies that don't involve drugs, that just involve measuring specific cells that are in the blood, specific cells in the respiratory secretions, in order to try to understand more about this virus. And then there are people working on mouse models of this virus and infecting the mice to try to understand if we can more rapidly test drugs that we already have, could be repurposed to treat this infection. And then my lab does, and it's working with other labs at UCSF, to study just the airway cells that are the first line of infection. So when you are exposed to this virus, it goes into your nose, mouth, or eyes. And one of the first cells that gets infected are those cells of your respiratory tract, the cells that line your nose, mouth, and lungs. And so we are testing, we grow those cells in Petri dishes, essentially, and we give them to the group that studies the virus, and they are testing a bunch of panel of drugs that are already available to rapidly screen any drugs that might inhibit viral replication.
That's fascinating. And that's great. I know we have projects also that are looking at the entirety of the immune system using some very, very fancy technology that allow people to get a snapshot of all of the different cells of the immune system to see what are the differences in people who have really, really severe COVID-19 versus those who have milder COVID-19 versus normal people, and to see what are the aberrations that appear to be driving a lot of this disease. There's some fascinating studies going on. We've had a few interesting questions asked here. First, somebody asked, is there more than one disease? Stacy Passman actually asked a good question. We asked, is there more than one disease that we're calling COVID? I'll answer that quickly and then sort of throw it back to you, Megan, and to Erin too as well. There's one virus causing COVID-19. I think there are certainly different clinical manifestations of COVID-19 related to the virus. I think there's the classical case where somebody develops a respiratory tract infection for a few days, then kind of seems to get a little bit better, and then develops more of a flu-like illness that kind of waxes more before it begins to wane after a few days, followed by like the sudden onset of like this catastrophic multi-organ system illness and acute respiratory distress syndrome that puts them in the ICU and on a ventilator. That's sort of the classicals, but within that, there are variations on the theme. There are people with mild disease that have a cold, there are people who are asymptomatic, there are people who just have a fever and muscle aches, and there are people who present de novo with the severe disease. Some people develop thrombosis, develop clots in their blood vessels, and so there are multiple clinical manifestations, but primarily of a single entity, namely the virus. Any thoughts, Megan, about that? Or
Couldn't have said it better. I agree.
Thanks, thanks, Erin. Quick question.
Wait, can I intervene there?
Yeah.
I do want to say that a lot of, you mentioned how many patients have been treated at Stanford and how many patients have been treated at SFGH. We've treated about 70 or so. And like you, we've seen these typical manifestations, and we've been hearing reports out of New York, these more unusual manifestations. And I want to reassure people that these are things that we have seen with influenza as well. Things like stroke and heart attack. We have known for a long time that influenza increases your risk of a stroke within the first 14 days of having influenza infection. This virus is similar to influenza. It's just a lot worse.
That's a great point. That's an outstanding point. I think there's a question, Jerry Hertz asked a great question. What is the survival rate if you're put on a ventilator? I think it depends. It depends what data you look at. But if you want to talk about the Bay Area, you heard from Dr. Marmor. I think 2 have died in the entire Stanford healthcare system. I don't know how many of those, out of how many that were on a ventilator to begin with. There have been 0 who've died at the General, despite having 36 or so on a ventilator. And I think UCSF's had 2 who've passed away in total amongst everybody. So
yeah, that's right. One of them was like a San Francisco General patient who was transferred over.
Oh, got it. Okay.
But yes.
They're massaging the data. What else is new?
Yeah.
Sorry about that.
But yes, I mean, the way we have interpreted this, there are lots of people speculating why the Bay Area has had lower mortality rates than other places in terms of ventilated patients. And there's lots of speculation. People have suggested that there might be a more virulent strain that circulated in Europe and then was transferred into New York, whereas here on the West Coast, we have largely been seeing patients with a strain that was introduced from China directly before going through Europe, that they're calling that the Washington strain. However, it should not, it needs to be emphasized that many of the colleagues reporting back from New York are describing a situation in New York that is not the standard of care. And we know that when you cannot give the standard of care because there are too many patients, that patients have a higher mortality. And one of the luxuries about living in California is that we have a governor that listened to the public health experts very early and sheltered in place, shut down schools about a week before other locations around the country. And our hospital systems shut down elective surgeries. And that resulted in us having a capacity of about 50%. So we had lots of doctors, lots of respiratory therapists, lots of nurses. We had everyone on board to take care of these patients. And that isn't the case in New York City, where people were dying in the emergency room without being seen. It was really, really, really horrible.
So I think that's a problem. I mean, that answers actually one of the. Let me make sure I'm unmuted here. Great. That basically answers one of the questions that I was gonna ask that were being asked here on the chat line, was basically, are there multiple strains of this causing different disease? And I share your point of view, Erin, exactly. I think a lot of it has to do with the fact that New York is one of the densest places on the planet, where everybody takes public transit, and where I frankly think they should have been shutting down the city before the first infection. Not after the infection had already taken hold, where you knew that it had been 2 weeks already of probably multiple infections spreading in this very, very dense city. And I think that is exactly why it overwhelmed the healthcare system, and people were not able to provide probably the best of care, and people were probably dying at home at other places, or getting to the hospital too late. There are, in fact, well, just so people know out there, according to Stanley Perlman, who's a very well-respected virologist at the University of Iowa, you know, it doesn't appear that this virus is mutating very fast. It certainly isn't to its advantage to mutate. It's doing a pretty darn good job spreading around the world as it is right now, relatively asymptomatically in many people. So there aren't a lot of mutations. There is some evidence that there are probably 3 variant strains, at least a minimum of 3, and that there may be a different strain in Europe than which affected New York more so than here. But like I said, it's not to the virus's advantage to become any more virulent than it is. It's probably to its advantage to become less virulent and to become part of the community with the rest of us. So if anything, further mutations would probably render it less probably virulent than it currently is now. But I agree. I think the primary things that we're seeing now in differences between various cities relates to the density of population and the degree of social distancing that was put into place, I think, early on. Okay, so with that, I just want to ask one more thing of Dr. Marmor, and then I'll get into a few other questions here at the end. Can you talk a little bit that little things do make a difference, right? I think it's been noticed by people who see a lot of this disease, for example, that the position somebody's body in actually helps with their breathing and helps with the amount of oxygenation they're able to get through their lungs. Even when their lungs are full of fluid and full of inflammatory debris. What have we learned about that? I've read that patients are actually being ventilated, or even prior to being ventilated, are being put on their bellies, prone, and that they actually breathe better when they're sort of in a swimming position, like they're doing the freestyle stroke, than when they're sitting up in a chair or lying backwards. Is that true? And what has been your experience?
Yes, so I'm a big believer in proning. We've known for a long time that for acute respiratory distress syndrome, when lungs are very, very inflamed and are having a hard time exchanging gas and getting oxygen into the blood and carbon dioxide out of the blood, that patients do really well when you turn them onto their stomach. It can feel, when you're short of breath, you can imagine it probably feels really good to sit up. So you can use all these muscles here in your chest to take bigger breaths, but sometimes that's not quite enough. When a person is sitting upright, a lot of the times the air they pull sits up here, mostly in the top of their chest. But unfortunately, when you're upright, all of the blood that needs to meet up with that air is going down to the bottom of the chest. So the two aren't overlapping. You have air going here and you have blood going down here, and they're not mingling enough. So we find that if you put someone face down in that prone position, almost like a swimmer, if you can imagine, doing sort of like a freestyle movement, you can imagine that one, you don't have that gravity preferentially moving all the blood to the base of the lung. You sort of eliminate the effect of gravity, and now you have blood sort of distributed more evenly throughout those lung fields. You can also then utilize, when you're breathing, you won't necessarily favor the top part of the lung here. You'll favor more even distribution of that air throughout your lungs so that there's more mingling, like we were talking about. The other thing to keep in mind is you actually have a lot more lung back here in your back that is otherwise going to be squished by your belly and your heart when you're laying on your back. So you can utilize more of those areas of lung when you're on your stomach. And so I'm a lover of proning. We've been proning people early, whether they're on the ventilator and whether they're not. The other thing it can help with is if you have a lot of secretions and phlegm stuck in the chest, it can be really easy to bring that up and forward when you're leaning on your stomach rather than upright.
That's great. Thank you very much. So that's about as good an answer. That, just so you guys who didn't go to medical school, that's was something I learned called the zones of the lung that I never understood in medical school. They were drawn and diagrammed for me, and I was tested on them about, you know, where blood flows and where air goes, and matching oxygenation to perfusion, as we call it. I never understood any of that. I'm not a good physiologist. So thank you. I now understand it for the first time after 25 years. That's how good you are. All right, so we've got a bunch of questions about the immunology of this disease a little bit as relates to drugs, testing. There have been questions about the strategies of allowing everybody to get infected and having herd immunity and stuff. So let me just address a lot of these questions here in the last 5 or 10 minutes here, and then we can take a few extra questions probably here at the end. So somebody asked a question about the strategy in Sweden. There has been a general strategy in Stockholm, primarily within Sweden, to basically allow the infection to run its gamut, to leave everything open and to not particularly socially distance, with the hope that there could be herd immunity, that there will be enough asymptomatic people that will spread the infection around, that people will develop an immune response to the virus that will end up protecting the population. And what do I think about that? And has Sweden really achieved herd immunity? Most people do not believe that Stockholm has achieved herd immunity right now, that the magical number where that would be achieved would be you have to have 60 to 80% of the population theoretically immune, if that's possible with this virus. And we don't even know that for a fact or a strategy. But Sweden has a lot of advantages over New York City, for example, and it's very possible that the UK also started with an initial strategy of at least experimenting with the fact that should they allow herd immunity to take effect and maybe protect the population that way. And very quickly, prior to Boris Johnson himself getting the virus as a result, I think the epidemiologist in the UK figured out that there would probably be in excess of multiple hundreds of thousands, if not 1 million deaths in the UK if that were allowed to happen. So Sweden's kind of a unique place. Whether there truly is herd immunity developing there or not, I think is not really known. And I think most reliable public health officials, and we'll deal more with this next week when we talk to Dr. Faust, who's really an expert in this area, about whether or not allowing herd immunity to sort of take place naturally was really an effective strategy or not. Ditto, we'll talk next week a little bit. There's some questions about when this virus began. There have been reports that somebody died in Santa Clara County at the beginning of February. I know for a fact that I had a patient infected at the very end of January. I have pretty strong circumstantial evidence of that. Yes, the virus was here early. And I think Dr. Faust can again talk about that and about what that means in terms of implications for public health and the origins of this whole infection in the first place. So let me end by talking about a few areas where I think people have a lot of questions. First of all, testing. How do we test for the virus? The virus is essentially what we call an RNA virus. Like I alluded to earlier, the genetic material in all living beings that is who we are, and that when our cells replicate, drives that replicative process, is DNA. And this particular virus is made up of RNA. So what the virus has to do is it actually has to get inside, bind to and get inside the epithelial cells in the nasopharynx or the eyes or the oral tract or wherever. And then it has to replicate itself, and it has to do so by basically hijacking the host machinery to basically drive what we call transcription of the RNA to make proteins. And in some respects, there are various enzymes that can make, for example, with HIV, that can make RNA into DNA, and there are other enzymes that can drive RNA to be converted, to be translated, as we say, into proteins as well. And there are various drugs that are out there that can block those various processes. But the way that we test, for the virus is we actually have to detect whether or not there's RNA, viral RNA inside of a person. And so you take a very uncomfortable swab. I've had this done. Have you been tested, Meghan and Erin, at all for this? Nope. And any of you tested? So I have. I've been tested. It is not a pleasant experience. You take a swab that's about that long with a very rough tip that's got little brushes on it. And it's basically jammed up your nose all the way back. I thought it was behind my eyeballs. I thought they were going to rub through my cribriform plate into the back of my brain, basically. But they basically scrub very violently in all different directions for about 10 seconds in each nostril behind your eyeballs. Yeah, and it's quite. It left me crying basically when the procedure was done, just naturally tearing. But what you do with that swab is you then transport it to a lab. And in the lab, you need to figure out whether the RNA is there. And so there's a whole host of reagents and enzymes that are done to basically take that RNA and reverse, what we call reverse transcribe it into DNA. And then once it's then converted into DNA, there are a further reaction that can take place that can amplify the DNA. And if you in fact can detect amplified DNA from that sample, it's very, very specific for that genetic code, that RNA code that's in that virus. And so what we call an RTP, a reverse transcriptase PCR test, is the gold standard for diagnosing the virus. Now, it comes with a lot of pitfalls. First of all, if somebody doesn't swab you aggressively or misses an area where there's virus in that particular area, it's possible you'll shoot a blank. And you know, the reverse transcriptase reaction won't find any RNA to make into DNA, and therefore you won't get DNA, and it'll be a negative test. But you just missed it because you didn't swab the right area of the nose. And I think the false negative rate can be anywhere between you know, 10 and 30%, somewhere in that range. It's probably not 30%, and it's probably not as low as 5 or 10%. It's somewhere probably in between. You can also have false positives, and this gets into well, not really false positives, but like I alluded to, a virus is not a living organism per se. It's a collection of RNA and proteins. And the real question is whether or not that virus is infectious or not. So it's very possible, for example, that if somebody's been infected with the virus and cleared it from their cells, and those cells are being extruded from the body and being cleared by cells of the immune system, that there's viral debris that's left over. It's not intact virus, but there may be some residual RNA that hasn't been completely degraded yet. And a PCR test an RT-PCR test is so sensitive that it will amplify that, and it'll show up just as if somebody had the viral infection. So you can also get some quote unquote false negatives. It's not a perfect descriptor, but it's a good enough test, which will look like somebody's virally infected because you're detecting RNA, but they're not necessarily infectious. And I think a lot of people have asked, can you get reinfected again and stuff like that? And I think we don't know the answer to that, because a lot of people who have tested negative, who have been positive, who then tested negative, have subsequently retested as positive again. But you know, a few days later or a few weeks later, when they still may have debris left over from the carnage that ensued, that was part of their infection. And you really need to wait months to years when you re-swab someone to figure out whether or not, because then you shouldn't expect to see RNA debris left around, and then you will truly have a better idea whether or not these false positives are false positives or not, and be able to figure that out. What about antibodies?
Yes. Jonathan, I just wanted to interrupt real quick because I said the information is changing so rapidly. Just today published onto bioRxiv, so not peer reviewed yet. Yale posted the results of their saliva test and how it compared to the nasopharyngeal swab, RT-PCR, and it was better at detecting the virus in the saliva. And one of our main limitations right now in the United States is getting those swabs.
Yes.
So this may, in fact, free up our ability to do a lot more tests. We've been collaborating with Stanford. Stanford has a testing facility. We have a testing facility to try to increase the number of tests that we can run exponentially. We have not reached capacity. There is still more capacity. We can do more tests. We can run that PCR. We are just lacking in those swabs.
Yeah, and what's amazing, it's an excellent point. The saliva testing will make it possible to actually go out into the community and really do this on large scale, I think, in a much bigger way. I will also say it's not just that we have the capacity to test as well. The labs that we have at UCSF, and I'll even give a huge plug in here to the General, which I'm going to talk about here in a second. Not only have we come up with our own home brewed testing and have been able to develop not only the tests, but the capacity to test, that's in an amazing way, but it's also the turnaround time. The turnaround time at UCSF, we are now testing every patient being admitted to UCSF, to Parnassus right now, or going for an ambulatory procedure for the virus, to protect the healthcare workers. And we're able to do this because some incredible lab-based infectious disease docs have developed a turnaround time of between 1 and 2 hours for this test, basically, using some very fancy machinery and some very dedicated personnel. And so the ability to test everybody and test everybody within an hour or two and get your answer and not wait 48 hours or 72 hours or weeks sometimes in other cities is truly going to allow us to be nimble enough to contain this infection, at least within the hospitals and potentially elsewhere if there's a saliva test. So I think that's really, really amazing news, and I didn't know that. Regarding antibody testing, we at the General developed I know Stanford has had an antibody test for a couple of weeks. They've been using in the community. At UCSF, the first lab to come up with an antibody-based test for both acute infection and chronic and more longer-term more distant, remote infection was developed at San Francisco General with the immunology lab there that I think is spectacular. And so we've been able now for a couple of weeks to test for anti-COVID antibodies in our patients. And that tells us several things. First of all, for people who are unable to get swabbed or in whom we have issues with the false positivity or negativity, I should say, of the test, it allows us to actually surveil the antibody response in patients to see if they have what are called IgM antibodies to the virus, which would indicate a very, very recent infection. And then the immune system, when it goes on to sort of establish a more chronic response, antibody response to infections, converts the IgM response into an IgG response. And we can test for both forms of antibodies to the COVID-19 virus, and it's got remarkable specificity for this infection compared to other coronavirus infections and such. You may have read in the press that, that there's a whole host of different antibody tests that have been on the market, and many of them are pretty much unreliable. I will say that there have been studies now done at UCSF that were alluded to earlier. Somebody asked a question about the different, they read in the New York Times, I think Paul Sedway asked about all the different tests that he read about in the New York Times, and these were being studied at UCSF. And the answer is that yes, a lot of them are unreliable. They have cross-reactivity to other coronaviruses and such. But the tests that we have both at Parnassus now and at the General, and I believe at Stanford too, are what we call ELISA-based tests. They're blood-based tests that use a complex series of what we call enzyme-linked reactions that are very specific for this particular virus. And it's going to allow us now to look at the broader population as a whole to figure out who's been exposed, how much immunity possibly is there or not, and whether or not public health measures still need to be put into place. The last thing I want to talk about are vaccines. Everybody's been asking about treatments and vaccines, and maybe I'll save treatments for next week, except to say that there's a very exciting clinical trial that was announced by Dr. Fauci today that everybody's been sending me emails about. Is a drug called remdesivir that interferes with the viral replication process that I sort of alluded to earlier. I will just say that, yes, this is the first positive trial. Yes, it's going to be FDA approved on an accelerated basis, it looks like. And yes, it will be available, probably being able to be widely distributed, because Gilead is a very, very resourceful company that makes the drug. I have to say, looking at the clinical trial results, they're positive. It's not a panacea. And we can get into more of that. And I can maybe, if people have questions about that, we can talk about that as well.
Well, let me just say, I was super excited about this because I am a pulmonologist, right? You too, Megan. We were all excited about this. My friend was running the trial at Gilead, super excited. And if what Dr. Fauci said is true, based on the numbers that he reported, it had a modest effect. What has a really big effect on mortality in this disease, it seems like, is not having a surge situation.
Right, exactly. Bingo. I think that's the big message here. There's not going to be a drug that's going to allow us to sort of get out of this easily, I think. And that's sort of the last point I want to make about vaccines. Is there a vaccine coming? First of all, just because you have antibodies to the virus doesn't mean that those antibodies are protective, or what we would call neutralizing. There are plenty of people, as a matter of fact, the way we've diagnosed HIV in the 1980s and 1990s before modern molecular methods was we tested them for antibodies. Lots of people, right? Everybody with HIV made antibodies to HIV, but they weren't protected. People with hepatitis C make antibodies to hepatitis C. They're not protective. And furthermore, antibodies not only may not be protective, they can be harmful as well. One of the reasons why, for example, dengue fever, when it affects people, the first time someone gets it, it's a relatively modest disease, illness. The second time someone gets dengue and they mount an antibody response to the virus, there's been pretty convincing scientific evidence that suggests those antibodies enhance the viral infection and the immune response to viral infection, and people get very, very sick. And there have actually been vaccine trials that looked at vaccines to the original SARS virus in mice, and the SARS virus is like the granddaddy to this virus. They're both coronaviruses that are very similar. And in several of those experiments, depending upon what kinds of ingredients were put into the vaccine, some of the mice got very, very sick, what we call hypersensitivity reactions in the lungs when they were challenged with the virus itself. And the thought was that the antibodies derived from these vaccines that were generated when they met the virus created an immune response from these, what we call immune complexes that came together that actually drove what looked like an immune reaction in the lung itself. So you have to be cautious a little bit. And there's certainly been human clinical trials and other respiratory viral illnesses where some people got worse from the vaccine and not better. So,
Jonathan,
yeah.
Jonathan, I need to be the optimist here. I think we're going to have a vaccine, and I think we're going to have a vaccine probably this time next year. We'll be broadly immunizing people. And I could be wrong on this, but this is my speculation. The group in the UK that developed a vaccine for SARS that they never ended up getting into SARS-1, that they never ended up getting into a clinical trial because there wasn't enough disease, used the same methodology to develop a vaccine for SARS-CoV-2. It was safe in humans, and then they did a rhesus macaque monkey trial in which they infected they vaccinated the monkeys and then challenged them with SARS-CoV-2, high doses of SARS-CoV-2, and none of them got sick. It is starting its large clinical trial now for efficacy.
That's the Oxford
I am
group.
very, very, very optimistic. We will.
Yeah, I am too. I'm an optimist. I'm just laying out there. I can give you more thoughts, but I'm laying out there why I don't necessarily think it's as clear a path to a vaccine as people think that there'll actually be. One reason, Erin, just to be a devil's advocate to you, is if you look at people who actually had SARS itself, the majority of people who were infected with SARS, actually their immunity wanes. If you do if you both look for antibodies in patients as well as the B cell precursors and plasma cells that give rise to those antibodies, you can't find them in most patients when they're several years out from infection, beginning at 1 year farther out. So for some reason,
Jonathan, you know that it isn't just the antibody response that's important. You also especially if you give a live vaccine.
Yes, they do have a cellular immune response as well, but even the cellular immune response wasn't as robust, I think, as people had expected. But I'm just needing this to sort of say that there are a lot of factors that go into vaccine development, including both the safety of the vaccine as well as the long-term, short-term efficacy and long-term efficacy of the vaccine as well. And for reasons, vaccines for many viral infections do elude us. So I am 90% sure we'll have a vaccine just because there are 120 candidates out there right now, and many of them look good. J&J has a very good looking one as well. But I think just for people thinking that that's an excuse why they can go out now and sort of ignore social distancing and stuff, I think we're going to be changed as a society, and we'll talk about this more next week, for a long time to come. And hopefully we'll have that vaccine next year, maybe, that will prevent this from becoming a seasonal pandemic. Meghan, do you have any comments at all about this?
Yeah, I just wanted to say, I think that the reason that we're seeing such low numbers of death is because all of you, I actually want to just take a second to thank all of you for quarantining in your home. I mean, we're seeing the effect of that at our hospitals, and that's what's allowing us to take really good quality care of patients, both affected by COVID or by any other illness. And so, you're staying at home, minimizing trips out of the house, maintaining at least 6 feet from people who are outside of your household, keeping your noses and mouths covered when you leave your home. They're not for nothing. I would actually encourage each person to go online and take a look at some of the CDC recommendations on how to effectively wash your hands. I know that there's been, I've never seen a bottle of hand sanitizer since the middle of March to date. So if if all you have is just regular good old fashioned soap, making sure that you're doing the correct use so that you can rest assured that you've cleaned your hands effectively when you've come in and out of the house. All of those measures have made an enormous difference, and I want to thank you for that. I also share a lot of the skepticism that you've heard on this call about, we're trying to gather information, we're trying to understand this virus, and we're trying to understand how we're going to get to the other side of this virus. And I'll be honest when I say that I am personally budgeting in my mind a minimum of 2 years before I feel like society has sort of hit a stride of some sort, whether it includes a vaccine or not, in how we cope with COVID-19. So in the meantime, I do want to encourage all of you to maintain your health as best you possibly can. You know, like you've heard what Erin was saying, our numbers in the ER are very, very low. The way that we've reorganized our clinics and our hospital is such that we want to continue to protect you. And we believe that we've been doing a very good job of protecting ourselves as staff and protecting you as patients. So, you know, if I would not put off your other healthcare needs or allow those to fester in the background in favor of this pandemic right now.
So let me just add a little point. There was a question asked that gets just at what you were talking about, and I'll just do this before we sign off here. Somebody asked a question, actually Rita Jeremy asked a question about N95 masks, and specifically the one that she wears that has a valve at the end of it. We're not allowed, Rita, to use those masks in the hospital. And the reason they exist is because if you've ever worn an N95 mask for any length of time, it's not comfortable. I after 10 minutes with it on in a patient's room, I have to leave the room to take it off because I feel like I'm suffocating in it, because you can't breathe out as easily or in very easily through the mask. And so these masks were created with valves in them that let you exhale with ease, even though it protects you while you're inhaling. And so the masks actually will protect the wearer from particulate matter that's of a certain size from getting through the N95, but it doesn't protect people around you, which I think is what your question was, because you can exhale through that valve that's in the mask. So you're correct. If you happen to wear one of those masks and you're going out into the community and you want to protect other people, you do need to wear a droplet protection on top of that. You wear it usually under it as well. But it's not the same as the traditional N95 mask that doesn't have the valve. So it's a good question, and it fits right along with what Dr. Marmor was saying about continuing to be vigilant and which is doing more than anything to help all of us here in the hospital. So with that, Jessica, you want to sign us off? Thank you both, by the way, again, so much. You were the best.
So by way of closing, I just want to say a couple of things. First of all, as I scroll through the nearly 200 people who are on this Zoom call, I recognize so many people who are frontline workers, medical professionals, other people who have been making our society over their generations of contribution work. And so we're so proud of all of you and so grateful for everything that you've done. So I just want to say that I see you here, and I recognize that so many of you are a part of our healthcare system and other ways that our society functions, critical workers. Next week at the same time on Wednesday from 5 to 6 o'clock, John will be interviewing another doctor, Dr. Jeremy Faust, who grew up at Congregation Sherith Israel, who is the son of Leland and Susan Faust, who is a public health doctor, an emergency room doctor at the Bragg in Boston, and we'll have a chance to ask many of the questions about public health issues that have come up today. We are accepting questions in advance, so please do submit those to Peter Bonos if you would like to have Dr. Faust and also John see those in advance. I want to say thank you so much to our amazing doctors who have been on here, and to recognize that Meghan and Andrew have come to be a really important part of our congregation. We're thrilled to have you in every capacity in this congregation, and thank you so much for being with us today. Erin and Stephen, and I think it was Ben who ran into the room for just a minute there. I want to say we love your family and your kids. We've named them here, and they're part of our studio program. And I was delighted to see Ben. I'm sure you weren't, but I was delighted to see Ben for just a minute in the bedroom, wherever you are. So thank you so much to Dr. Marmor, Dr. Gordon, Dr. Graf, to all of you for helping us understand this really complex virus and this complex situation. And our Sherith Israel community continues to be here to support one another, to educate one another, and to be together even in this time when we're all in different places. So thank you very much. We look forward to seeing you throughout the week, Friday night at 6 o'clock. Please join us for Shabbat, 9:15 on Saturday morning for Torah study, and many other programs. Check our website next Wednesday, 5 to 6 p.m. right here. Stay healthy. Thank you all so very much.